problems. Hydrophobicity scale was the next big one. That is like a 50-second video. I highly recommend taking 50 seconds and watching it. Okay? The key thing here is that the hydrophobicity scale tells us an energetic cost, change in energy, positive or negative, to move any given amino acid from water to a hydrophobic environment. So that could be an apolar solvent or moving it into the bilayer. Stuff that is nonpolar wants to go from water to something nonpolar. That would be a net negative change in energy. That's energetically favorable. That wants to happen. Stuff that's charged, taking it out of water, out of something polar, and putting it in the membrane would be positive change in energy. Doesn't want to happen. Not energetically favorable to take something polar out of water and move it somewhere nonpolar. So that's kind of what's being shown here. Our charged residues have a positive change in energy score when you move from water to the bilayer. Our aromatic stuff this is kind of all the things in the middle that are sort of, some stuff is really, it doesn't matter. But then we have stuff that's really big and aromatic, very nonpolar. Putting it into the membrane is very favorable. We have a negative score. Okay? The scale's a little bit different on here. The scale on there is shown in kilocal per mole. What you will have on your reference sheet, which did everyone get one? I passed out the reference sheets. If you don't have one, are there extras? Who has extras? Hand them to people who don't have one. I want to save trees. Please bring this back on Monday, okay? If you don't, there will be some copies, but hopefully your teammates remember them. I'm also going to let you write on the front of it, so you want to bring your own back, okay? We'll talk about that more in a second. So don't lose that, use your reference sheet. But what you can see on there is I'm giving you the scale from the textbook, which looks like this, okay? Color coding is the same. Blue means hydrophobic on this plot I grabbed from the internet. Red means hydrophilic. The scores in terms of actual number are a little bit different. I don't need you to memorize these numbers. I need you to understand negative versus positive and what this energetic cost is. Yes? Why is tyrosine? Probably size. Tyrosine has, in this aromatic ring, it has a ring structure. Yeah, so again, it's not going to perfectly match sort of the scale of just looking at which ones are polar, which ones are charged, which ones are which. So yeah, that's a good question. But this is, interpretation here. And tyrosine, remember, this is a literal study that someone took. Tyrosine on here falls closer at a little bit. It's not quite as far in the aromatic region. So the hydrophobicity plots, what can we do with them? Spoilers, it's kind of the same as Ramachandran. It's kind of the same as everything. We want to figure out protein structure. We're trying to predict protein structure. We're trying to design protein structure. We're trying to figure out where the proteins function, where they might be located, what they do. Location and function are dictated by the structure they have. Okay? So the hydrophobicity plots can help us figure that out. So in this one here, these negative scores, again, and this, you can see this scale is hugely different now, okay? Don't worry about score in absolute value. Worry about sign and directionality. So stuff that is very hydrophobic is up here, okay? So if you imagine going down your protein chain from 0 to 300 amino acids and plotting the hydrophobicity score or scale for every single one as you go, so you have hydrophilic stuff and you kind of switch to hydrophobic stuff. Hydrophilic stuff's on the inside or outside of this membrane. The hydrophobic stuff is traversing the membrane. So in order to have a transmembrane segment, you probably need at least 20 amino acids in a row. You need them to be sufficiently hydrophobic, okay? Stuff that's kind of bouncing around here wouldn't make sense as a transmembrane segment. Now, for this one, it's showing you that this is, this one might be the bacterial rhodopsin, I can't remember. That this one has roughly these 5 transmembrane alpha helices in there, indicated in yellow. They're the 5 big hydrophobic peaks of sufficient length of amino acids. I can't, from this alone, without this figure, know if this is an alpha helix. It could be a beta sheet. What I can say is it's sufficiently hydrophobic, and it's a long enough string of hydrophobic stuff that it could get across the membrane. So this is, I could predict that this could be a transmembrane segment. Okay? I can't say definitively. The other thing is that if I don't know that the location is transmembrane, if I see something like this, I might predict that I have some buried helices, okay? And remember, I'm not going over helical wheels again, okay? You will see one, have to interpret it where it could go. You won't plot one on the exam, but be prepared to read one, okay? And so again, from just a plot of hydrophobicity alone, I can kind of say if I think something could be transmembrane. I could say if it's sufficiently hydrophobic, would it be in the core? If it's a globular protein, there's a lot of things I could say. I don't know definitively. Okay? I'm not asking you to be able to speak in absolutes, but it's about helping you predict. If you know some of these things, if you know location because you know where you isolated it from, well, then that helps you get further along in structure and function. Okay, other questions about hydrophobicity, scale, index, what that sign means.